The impact of water source on plant growth is what our final project is about. It's a comparative study using the Coleus plant. This project focuses on the effects of different water sources have on plant growth. The Coleus plant was chosen for this because it has easy growth and germinates quite quickly within 10 to 15 days. Four Coleuses were planted in the same soil with similar conditions and water using two different bottle types. Fiji water, Brita, East Spring Street tap water, and Evan Scholar tap water. The height of each plant was monitored over time. Once they started growing after about 2 weeks, only the Brita plant was growing, and over spring break, the Fiji plant started to grow. So these were the two plants that were growing, shown in the bottom left corner. And through this study, we investigated the different types by maintaining the environment to be quite the same, like I stated. And now, Jared.
Um, so of the four plants we had growing, two of them you could probably consider successes. Two grew nominally, reaching heights of nearly 7 centimeters, and two sources of water really struggled to grow. I attempted on 3 occasions to replant, and even used radish seeds at one point because of their quicker germination time, and still could not get anything to grow under the 18 East Spring Street tap source. And this could be due to a variety of reasons. And Maven will go over the methodology in our study.
So for our methods of testing, we used four different water sources to water each plant. We used testing strips to test the pH and mineral contents and tested each water source at least twice in order to make sure that each source was consistent throughout the experiment. The Fiji and Evans Scholar tap water had the same hardness of 25, and the Brita and the East Spring Street had the same hardness of 120. So while there were similarities in the water sources, the amount of each kind of minerals they had were different enough that we could use them to understand the differences each water source had on the growth of the plants.
So, looking at our results, we observed that the Brita filtered and Fiji bottled water consistently supported healthy plant growth, while straight tap water from two local sources, like we mentioned, resulted in no measurable growth. The difference appears to stem from the water quality factors that directly affected soil chemistry, um, and plant nutrient uptake. The tap water showed high alkalinity levels around, higher levels than we saw otherwise, which likely raised soil pH and hindered the availability of key nutrients. Additionally, elevated hardness caused by excess calcium and magnesium, like seen at the Evans Scholars tab, may have interfered with the absorption of other essential nutrients, creating some sort of imbalance with the soil and the plants. The sodium chloride concentration in the tap water was also high, particularly at Evans, which could have contributed to root level dehydration and impaired water uptake due to osmotic stress. At the Evans Scholars location, medium iron levels were detected. While iron is necessary in small amounts, too much can lead to toxicity, especially when combined with other stressors like high pH or salinity. In contrast, the Brita filtration reduced hardness and removed interfering minerals, while the Fiji water, known for its balanced composition and low contaminant levels, offered a plant-friendly profile that promoted optimal growth. These results suggest that filtration or selecting water sources with low mineral content can significantly improve plant health by maintaining a nutrient accessible and non-toxic growing environment.